Hello dear students, I am Dr. Moin. In this video we are going to discuss about chromatography. This is the first lecture about chromatography. So in this lecture I'll try to make you people understand about the basic terms of chromatography. So you need to watch this video till the end. So here are the contents of current video. We'll see the introduction of chromatography. Then we'll see its basic types, which are column chromatography and planar chromatography. We'll see the phenomena of elution. We'll see what is chromatogram. Then we'll see what is distribution constant, and in the end we'll see what is retention times. So let's start with introduction. In 1901, the Russian botanist Mikhail Tswett invented adsorption chromatography during his research on plants' pigments. So this technique was invented in 1901 by Mikhail Tswett. And what was he doing actually? He separated different colored chlorophylls and carotenoid pigments which were obtained from leaves by passing an extract of the leaves through a column, and this column was packed with a material which was composed of calcium carbonate, alumina and sucrose. And elution means separation was done using the solvent which was a mixture of petroleum ether and ethanol. So what did he actually do? He packed the column with calcium carbonate, alumina and sucrose, and by using this column he separated various chlorophylls and carotenoids which were present in the plant's extract, and he separated them by using the mixture of petroleum ether and ethanol. Then one of his publication in 1906, he used the term chromatography for this technique, and he used this word by taking it from the Greek words chroma meaning color and graphos mean to write. So chromatography was taken from the Greek language. Chromatography is a widely used method for the separation, identification and determination of the chemical components in the complex mixtures. So this can be used as a definition of chromatography. And these days, no other separation method is so much powerful and applicable as that of chromatography. IUPAC has drafted a recommended definition of chromatography, which is given over here, and that is, chromatography is a typical method of separation in which the components to be separated are distributed between two phases, one of which is stationary phase while other is mobile phase, which moves in a definite direction. Means the sample or the mixture of the sample which is to be separated, actually that is distributed among two phases, and these two phases, one of them is stationary phase while other is mobile phase. Now, the stationary phase in the chromatography is a phase which is fixed, so it remains stationary. It is fixed in place, and it is present either in the form of column or on a planar surface. And the stationary phase, this may be solid or it may be in liquid form. Then mobile phase. So the mobile phase in chromatography is a phase that moves, means which is mobile. So it moves over through the stationary phase, carrying with it the analyte mixture. So mobile phase moves over the stationary phase and it carries the analyte mixture, means our sample, and in this way the separation occurs. And the mobile phase may be a gas or a liquid, or it may be a supercritical fluid. The term chromatography has been applied to several systems and techniques. So there are various types of chromatography. And all of these methods, however, have in common the use of stationary phase and mobile phase. Mean whatever type of chromatography we are using, there is always a stationary phase and a mobile phase. Components of a mixture are carried through the stationary phase by the flow of a mobile phase, and separations are based on differences in migration rates among the mobile phase components. What does this mean actually? Actually, mobile phase carries the components of our sample over the stationary phase, and there are different components in our sample, and each have different affinity towards stationary phase and mobile phase. So the component which has more affinity towards stationary phase spends more time with stationary phase, while the component which has more affinity towards mobile phase spends more time towards in mobile phase. So in this way, all of these components are separated from each other. Then chromatographic methods are of two basic types. So there are two basic types of chromatography: column chromatography and planar chromatography. In column chromatography, the stationary phase is held in a narrow tube. So packing of the stationary phase is done in a narrow tube which is called column, and the mobile phase is forced to move through the tube under the action of pressure or under the action of gravity. While in case of planar chromatography, the stationary phase is supported on a flat plate or it is present in the pores of the paper, while the mobile phase moves through the stationary phase under the action of which is called capillary action or under the influence of gravity. So now we will see various column chromatographic methods. Column chromatographic methods fall into three categories, which are based on the nature of mobile phase as shown here. So first one, in which the mobile phase is a gas, and this technique is called gas chromatography. And the method, it has further two types, which is based upon the physical state of stationary phase. So if the stationary phase is liquid adsorbed or bonded to solid surface, then the method is called gas liquid chromatography, GLC. And what is the type of equilibrium there? So it's a partition. So whenever the stationary phase is in liquid state, this type is called partition chromatography, and when it is in the form of solid, so it is also known as adsorption chromatography. So equilibrium there is the partition between gas and liquid. And the second type, in which the stationary phase is in solid state, so this is called gas solid chromatography, GSC, and the equilibrium type is adsorption chromatography. So in both of these types, the mobile phase was gas. Now the second type, in which the mobile phase will be in liquid state, and this is called liquid chromatography, LC. And there are further 5 types of it, means different stationary phases can be used over here. So if stationary phase is liquid, adsorbed or bonded to a solid surface, then this method is called liquid-liquid or partition chromatography. So definitely the equilibrium will be partition between these two immiscible liquids. Then the second type, in which the stationary phase is solid, so this is called liquid-solid or adsorption chromatography, and the equilibrium is adsorption there. Then the third type, in which the stationary phase is an ion exchange resin, so also known as ion exchange chromatography. So equilibrium type is again the ion exchange. The next, in which the stationary phase is liquid in interstices of polymeric solid, so this one is called size exclusion chromatography, in which particles are separated from each other on the basis of their molecular sizes. So equilibrium there is partition or sieving. And the next type, in which the stationary phase, that is the group specific liquid bonded to a solid surface, and this is called affinity chromatography, and the equilibrium there is partition between surface liquid and mobile liquid. So these are the 5 types in which the mobile phase is in liquid form. So these are the type of liquid chromatography. And third type in which the mobile phase, that is some sort of supercritical fluid. So the stationary phase could be the organic species bonded to a solid surface, and the equilibrium there will be the partition between supercritical fluid and the bonded surface. Now we will see the planar chromatography. It includes different types like thin layer chromatography, paper chromatography, and electrochromatography. Each makes use of a flat or relatively thin layer of material that is either self-supporting or is coated on a glass, plastic, or metal surface. So actually we are talking about stationary phase here, which is present in the form of thin layer, either self-supporting or it's coated on some support like glass, plastic, on metal surface. While the mobile phase moves through the stationary phase by capillary action, or sometimes it may be assisted by gravity or an electrical potential in case of electrochromatography. Planar chromatography was once called two-dimensional chromatography, but now this term has come to signify the coupling of two chromatographic techniques with different separation mechanisms. Most planar chromatography is based on a thin layer technique I talked earlier, and it is much faster, has better resolution, and it is much more sensitive as compared to its paper counterpart, that is paper chromatography. Now we will see the elution in column chromatography, that is the process of separation of various components in column chromatography. So elution is the process in which solutes are washed through a stationary phase by the movement of a mobile phase. So this process is called elution, in which the solutes, that is the various components present in samples, so these are separated through the stationary phase by the movement of a mobile phase. While another term which is used, that is eluent. An eluent is a solvent used to carry the components of a mixture through a stationary phase. So eluent is the solvent, or you can say the mobile phase, which actually carries the components of mixture through the stationary phase. So here is the figure which shows how two components A and B of a sample are resolved on a packed column by elution. So we can see here is the column and this is a packed column, and here is our sample, which is actually the mixture of two components A and B, and we are going to separate both of these components through this column. The column consists of narrow bore tubing that is packed with a finely divided inert solid that holds the stationary phase on its surface. So here we can see this is the column, it's a narrow bore tubing, and inside it is packed by the stationary phase, which may be solid or liquid, and here the sample is introduced, which is a mixture of two components A and B, and then we make to flow the mobile phase, which carry these components through the column, and both of these are separated gradually. Then isolation of components is then accomplished by passing a sufficient quantity of mobile phase through the column to cause the individual bands to pass out the end to be eluted from column, where they can be collected or detected at different times. So we can see here's the column which is packed by stationary phase. We have introduced the sample, which is the mixture of two components A and B. Then we used mobile phase, that is solvent, to make flow of these sample components through the stationary phase. So here is time 0 and now the process starts. So here at T1 we can see the sample has reached over here and the components are separated from each other. Then at the next time T2, these are further moved ahead and they are well separated from each other. Then at time 3, T3, they are further moved ahead, and so on. And then we have attached a detector with this column. So you can say at these three times T0, T1 and T2 there is a baseline, so there is no peak because no component has came out of the column yet. But at T3 A has came out of the column, so detector has given us the result in the form of peak, which shows that the A component is present here. And then at T4, at time T4, B component is out of the column, so we got another peak describing the component B. Now we will see what is chromatogram. So if a detector that responds to solute concentration is placed at the end of column as shown earlier during elution, and its signal is plotted as a function of time or of volume of the added mobile phase, so we get a series of peaks, and such a plot is called chromatogram. So actually the final result obtained after elution, that is called chromatogram. Chromatogram is useful for us as it gives us information, both the qualitative and quantitative information about our sample. So the position of the peaks on the time axis, so these give us the information about the qualitative analysis, that is which components are present inside, so we can get it by the position of the peaks, while the area under the peak that give us the information about the quantitative analysis, that is in what quantity that component present in our sample. Another term which is frequently being used in chromatography is the distribution constant. So all chromatographic separations are based on differences in the extent to which solutes are distributed between the mobile and stationary phase. So actually during the elution process, solute distributes itself among the two phases, which are stationary phase and mobile phase, and their concentration in these phases actually give us this distribution constant. For example, for the solute species A, the equilibrium is described by the equation, that is the concentration of this A in mobile phase and what is the concentration of this A in stationary phase. So equilibrium has been established, so now the equilibrium constant Kc for this reaction, and that is actually called distribution constant, and it is defined as, which is given in the form of equation, that is distribution constant, which is equal to the concentration of that component in stationary phase over concentration of that component in mobile phase. So in this way distribution constant can be calculated. So the distribution constant for a solute in chromatography is equal to the ratio of its molar concentration in stationary phase to its molar concentration in the mobile phase. Another parameter which is used in chromatography is the retention times. A simple chromatograph of two component mixture is shown here. So we can see here is a chromatogram, and it's a two components mixture, which is obvious from the two peaks, this one and this one. The small peak on the left is for a species that is not retained by the stationary phase. It actually represents the mobile phase. So this peak actually represents the mobile phase, and this peak has been appeared after some time, which is represented here as Tm. So the time Tm after sample injection, and this time starts from the injection time. So the time Tm after sample injection for this peak to appear is sometime called, so this Tm actually it is called dead time or void time, and this dead time provides the measure of the average rate of the migration of the mobile phase. So as I mentioned earlier, this dead time, it gives us the information that in what time the mobile phase passes through the column, and it is an important parameter for the identification of analyte peaks, the second one. while the larger peak on the right side is that of analyte species, this one. And the time required for this zone to reach the detector after sample injection is called retention time, and that is given by the symbol TR. Means the time between the injection of sample to the time of its appearance towards detector. This is called retention time. The analyte has been retained because it spends the time Ts in the stationary phase. So analyte actually it has spent more time, which is Ts in the column. So the retention time that can be calculated as, so Tr is equal to Ts spending time plus its void time Tm. So it gives us the retention time. So the retention time Tr, actually it is the time between the injection of a sample and the appearance of the solute peak at the detector of the chromatographic column. So this is called retention time. So dear students, this is all about the part 1 of chromatography. Hope you understand it. So if you like my video, then like it, and if you didn't subscribe my channel yet, then subscribe it right now to get in touch with my upcoming videos. So thanks for watching. Thank you very much.